Well, thanks very much. I'm going to show primarily one, it'd be one CT scan, but I do want to say up front, I can attest to, although I have no corporate relationship with the company, we've been using it now for several years. And provided you have a high quality operator, like the person I'm going to introduce you to, it is a fantastic machine. Anyway, I'm going to talk about why people are so blue when they have COVID-19 pneumonia. And this is a story of a virus attacking the mitochondria. And if you're interested in developmental biology or in the origins of life, it might strike you that mitochondria, which you see here in red in this picture, were once bacteria. So this is like a virus on bacteria war that goes on when you have COVID-19. This is actually a pulmonary artery smooth muscle cell. Here's another one. There's the blue nuclei, and the red are mitochondria in these three cells. So just a little science being art. So let's go on to the presentation. So these are the people that did the work. And I'll just point out Elahe Alazeta, who's our excellent physicist who operates the system. And without her, we wouldn't get the quality pictures we do. And we have, I can't introduce all these people for time's sake, but Shay Kolpitz is our virologist collaborator. And then we have an amazing team in the lab that actually have made a lot of the constructs. Ashish Dasgupta, Kwong Hui Chen, who have been instrumental in this, and Jeff Newbert on the confocal microscope. So lots of other people I can't mention, but who contributed importantly. So today, and briefly, I'm going to tell you that this is the third pandemic that we've had with the coronavirus in less than 20 years, SARS, MERS, and now COVID-19. So it might be nice to develop an understanding of how this damages cells and figure out therapies in addition to vaccines. I'm going to show you a case briefly. I'll remind you that the major complications of this virus are cardiopulmonary. That there are few effective therapies, and I'm not trying to trash anyone else's therapy, but other than dexamethasone, which is beneficial, the benefits from most other approved therapies like remdesivir and tocilizumab are modest. Vaccines definitely work for 20% of the world's population is vaccine hesitant, and with variants like the Delta variant, there is a possibility of immune evasion. Again, there's a room for new therapeutics in this area. The idea I'm going to present today is that mitochondria in the airway epithelial cells and pulmonary artery smooth muscle cells and the blood vessels in the lung are directly targeted by the SARS-CoV-2 virus. And this mitochondrial damage, I'm going to try to convince you, contributes to the airway infiltrates by inducing airway epithelial cell apoptosis, programmed cell death, and by inhibiting a normal response that autoregulates blood flow in the lung called hypoxic pulmonary vasoconstriction, or HPV. And that enhancing HPV might be helpful. So this is a big problem. It's the biggest problem we face apart from one or two global wars. By mid-June, we had 173 million infections, 3.7 million deaths. And I made the slide a week ago, and that's gone up significantly. So this is a case from a friend of mine early in the pandemic, Dr. Will Sharp, at a place I used to work, University of Chicago, of a 60-year-old female who had a history of hypertension. She presented with shortness of breath, cough, diarrhea, runny nose, sore throat, and abdominal pain. She had been well until 2 weeks ago. She had a known COVID exposure from a coworker at the Illinois Department of Public Health. And when she was seen by Will in the emergency room, the most remarkable thing was that she was hypertensive, afebrile, but her O2 sat on room air was 75%, which is quite profoundly hypoxic. And his comment at the time was, she doesn't look as bad as her oxygen saturation and tachypnea would indicate she should look. So she had what they call happy hypoxemia. Anyway, she deteriorated. This is her chest x-ray. And again, this shows the bilateral infiltrate that you see in most patients with COVID-19, which is reflecting airway damage due to alveolar cell damage and fluid accumulation. You can see here that her FiO2 was 60%, but her O2, the PO2 in the arteries was only 68 millimeters of mercury. So she was profoundly hypoxemic. She was enrolled in a trial of remdesivir. Remember, this was early 2020. So what is this virus doing? And I'm not going to go into this in detail for time's sake, but when the virus binds to the ACE receptor and comes in, it's translated, makes a polyprotein, and that basically, it results in the direct production of non-structural proteins, and then there's a viral polymerase that replicates the viral genome, and it goes on and makes the structural proteins, which are processed within our own endoplasmic reticulum, so it hijacks our system, and then basically packages both the packages these structural proteins in a structure called the ERGIC, and then it exports the virus and the cycle continues. But I want to try to convince you that all these non-structural proteins and structural proteins that are involved in making new virus are also attacking by direct protein-protein interactions, as well probably as by creating inflammatory mediators, but I'm going to focus on the direct protein-protein interactions. They're attacking the mitochondria, and I'll show you that there is actually evidence for that crazy idea. So just a little reminder for those of you that aren't virologists, that I'm a cardiologist, so I would be one of those people. SARS-CoV-2 is an enveloped single-stranded positive sense RNA virus that's spread by droplets. The lung injury is due to diffuse alveolar damage, and I believe disproportionate hypoxemia reflects VQ mismatching, ventilation perfusion mismatching. So the virus itself, as we see in this cartoon, encodes 16 non-structural proteins that form a replicase transcriptase complex. And then there are 4 structural proteins, and you probably all know about the spike protein because that's the one that all the vaccines target. That's the antigen source in the vaccines. And then there are 9 accessory factors. And I'm going to show you that we study some of these non-structural proteins and structural proteins and their interactions with mitochondrial proteins. And for those of you that went to medical school like I did in the 70s or 80s, I graduated in 81. Mitochondria, you'll be happy to know, still make ATP via the electron transport chain. But we now know, and in my lab we study this, mitochondria have a very dynamic life cycle. They join together, which is called fusion. They divide, which is called fission. They have quality control processes like mitophagy. And every time a cell divides, as we see down in the bottom panel, there's the nucleus in red and the mitochondria in green. The mitochondria have to divide. That's called mitotic fission. And we've shown in our lab that if you block mitotic fission, you prevent cell division and cause cell cycle arrest. So the hypothesis is that SARS-CoV-2 causes diffuse alveolar damage and hypoxemia in part by damaging mitochondria in airway epithelial cells and pulmonary artery smooth muscle cells. So here's a cartoon of an alveolus, and you can see 2 different types of alveolar epithelial cells. And just imagine if these cells starting to growing virally induce programmed cell death, you get gaps in the epithelial membrane and infiltration of cells and leak of fluid from the capillaries. And one of the ways it does that I don't have time to talk about today is through an apoptosis inducing factor called APE, which we've shown this virus produces. Simultaneously, while it's causing the havoc in the airway epithelial cells, it's also inhibiting HPV. And you all have HPV. When you get pneumonia, like in your left lower lobe in blue, the blood vessels there constrict, hypoxic pulmonary vasoconstriction, diverting blood to where there's air, which is called VQ matching, ventilation perfusion matching. And one of the early observations in a patient like the one Dr. Sharp had was that there is an impairment of VQ matching. Blood is going down here and picking up hypoxia. It shouldn't go there, it should go up here. This is not just a crazy idea. In fact, RNA viruses are well known to attack mitochondria, and very, very briefly, through a mechanism that involves BID, which is an apoptosis factor, and APE, the mitochondria transition pore opens and the mitochondria depolarizes. And these little balls that you see are cytochrome C and apoptosis inducing factor and other mediators of apoptosis that come pouring out when the mitochondrial membrane potential depolarizes. So viruses attacking mitochondria is a known thing. Likewise, this homeostatic oxygen sensing system that I mentioned, which includes the carotid body, which senses whether you're hypoxic, and I don't have time to talk about that, but is that happening? Explaining the hypoxemia. Pulmonary vasoconstriction, when your airways become hypoxic, you divert blood to better ventilated airways. And the ductus, which we actually also image using that MILabs CT scanner, but I'm going to talk about that. The cool thing, though, is that HPV is sensed by the mitochondria. So this is work by our lab and others. So when you get hypoxic here, in the wall of the pulmonary arteries through muscle cells, what's happening is that there's a change in electron flux and radical production. So very much HPV is sensed by mitochondria. That's not controversial. And recent work by Kim Dunham-Snary in my lab and my friend Ken Weir shows that complex 1 is very important in the oxygen sensing. And the reason I mentioned complex 1 in this little subunit called MDUS2 is the virus attacks many subunits of complex 1. These are major targets for SARS-CoV-2. So that may be how it inhibits HPV. So early in the pandemic, we came up with this theory, which we published in Circulation. And we again show a cartoon. Here's the pulmonary artery bringing hypoxic lung. blood into the lung, and there's a mucus plug to this airway, so it's hypoxic. And so blood is diverted to a better ventilated area and oxygenation is maintained. In ARDS, acute respiratory distress syndrome, which is what COVID-19 kind of looks like, there's actually an increase in pulmonary vasoconstriction in the small pulmonary arteries, and pulmonary vascular resistance is elevated, and then inflammation causes fluid accumulation. Early in the pandemic, some people thought that COVID-19 looked like high altitude pulmonary edema, which you would get if you climbed up Mount Everest too quickly. But HAPE is actually quite different, as we pointed out in this article. HAPE is not only exaggerated pulmonary vasoconstriction, but it's heterogeneous in its distribution, and there's pulmonary venoconstriction, which doesn't occur in COVID-19. And here's what our idea of COVID-19 was, and it's proven to be fairly accurate. So we thought HPV was suppressed, and so what was happening is, despite the fact you had an infiltrate in this lobe, blood continued to pour in, and there was also an elevation in the afterload, and so you were getting both hydrostatic leak as well as inflammatory leak into the airways, and you didn't have HPV to better divert blood to the right zones. So is this true? Well, if you look back at the SARS epidemic caused by SARS-CoV, which is a related coronavirus, 36% of all upregulated human proteins and 19% of all downregulated human proteins were mitochondria. So when you infect with that virus, this is the change you observe. And you can see this virus, SARS-CoV, attacks the mitochondrial matrix proteins, ATP metabolism, cellular respiration, etc. So very relevant to oxygen sensing and to apoptosis. And then this recent paper by Gordon now looks at the SARS-CoV-2 virus, the actual virus that causes COVID-19. And they basically took 26 of the proteins from SARS. They found that they bound to 332 human proteins physically. So this is a direct interaction beyond what inflammation does. And several of those were mitochondria. So this is their process in brief. They use mass spectroscopy to identify 332 protein-protein interactions. So here's a subunit of this virus in red, and it interacts with this network of human proteins. And they found that where this most commonly occurred was in the lung, although it also occurred in other tissues. And in terms of what are these subunits on the horizontal axis here, all these various components of the virus, what are they attacking? You can see one of the biggest targets is the mitochondrial matrix, and to some extent, cell cycle transition. And interestingly, we'll talk about it, but the virus that most closely resembled this interactome was the West Nile virus, which is another positive sense single-strand RNA virus. So this is from their paper, and I just wanted to highlight all the mitochondrial metabolic targets, mitochondrial ribosomal targets, and electron transport targets. So these are things that will change the survival of cells in apoptosis, and in the case of the electron transport chain, could change HPV. And you see the red viral subunits that are interacting with all these human black proteins. So here's the work we actually did. First, we hijacked the HIV virus for AIDS and used the lentivirus to make a vector to transduce cells. And I thank Dr. Colfitz for educating me on this. So Ashish Dasgupta and Kwong-Hui Chen do this work. Here's a GFP reporter. We've infected some human cells with the lentivirus. And you can see over time, you get accumulation of the expressed protein that is from the virus. Now, no SARS-CoV-2 virus ever just gives the host one protein. So it's artificial, but it gives us precision for what the particular protein components of the virus do. So first, we just confirmed that when we give NSP9, non-structural protein 9, we can actually record it, that it's present. So it works, basically. Then we went on to see, well, what does this do to the apoptosis process using human airway epithelial cells called B2B cells. And you can see that compared to the GFP control, the non-structural protein 7 and 9 and the M protein, which is a structural protein, all upregulate this apoptosis-inducing factor. They also changed mitochondrial dynamics proteins like diamine-related protein 1, which causes fission. And uniquely, the M protein caused downregulation of cyclin-dependent kinase 4, which regulates cell cycle progression. So when we look at, is there evidence that the SARS-CoV-2 proteins upregulated apoptosis mediator, this apoptosis-inducing factor, which is shown in green? Here's the control GFP, and you can see a big upregulation of green in the mitochondria. These little dots are mitochondria. in the Bs2B cells. So all three upregulated the expression of APE relative to a reporter. Did that cause apoptosis? Yes, it did. Here's the GFP lentivirus, and you can see all three of the heterologously expressed SARS-CoV-2 proteins increased apoptosis. And uniquely, consistent with the CDK4 expression, you can see that only the M protein, which is the white bar, this is a cell cycle progression chart with G1, G0, S phase, and G2M phase. You can see that the M, when you overexpress the M protein, cells got stuck in G1, G0, meaning they couldn't replicate, and that was unique to the M protein. I mentioned mitochondrial dynamics. Here's the control cells that are uninfected. When we put in lenti-GFP, it doesn't change it much, but the M protein causes fusion. The mitochondria are more linear instead of being little particulate structures, whereas the NSP7 and NSP9 cause fission. What the net balance is of a live virus, I'll show you later. So then we move from apoptosis to does it inhibit HPV? And this is Rachel Bentley, who's a master's student, now PhD candidate in my lab. And she took human pulmonary arteries from muscle cells, and she used as a surrogate for HPV. She used the rise in calcium when these cells went from normoxia to hypoxia, like with pneumonia. And you can see calcium goes up in these cells. That's unique to pulmonary arteries through muscle cells. And when she made her mean data chart, you can see here the baseline calcium level, it goes up with hypoxia in the control situation, returns to normoxia in green. And if we give potassium chloride, of course, as a control, it goes up nicely. And all the viral proteins inhibited that pink rise in calcium, but they didn't affect the KCl rise. So they specifically suppress HPV. So on to the last little bit of this talk. We then went to a live virus, because up to now we're using components of SARS-CoV-2. And this virus is called MHP1, the murine hepatitis virus. But when you nebulize it in the nose of mice, it causes COVID-19 like pneumonia. So here's what happens in cells with the MHP1. You can see the mitochondria in these LA4 murine adenoma cells. There's the nucleus. And when you infect them with MHP1, you can see two things. First, the mitochondria become more fused and lengthy, suggesting the net effect of this virus is fusion. And the other thing, this is a potentiometric dye called TMRM, tetramethylrhodamine. And when the mitochondria depolarize, they turn darker color. And you can see the loss of membrane polarity with infection, just like in the cartoon I showed you. And this is the precedent for subsequent apoptosis. So are they getting release of APE? Yes, they are. Here's the control green APE. Here's the infected cell with all kinds of apoptosis inducing factor. And you can also see APE has moved from the mitochondria into the now green nucleus compared to the blank nucleus in control. So finally, I know MILabs is probably saying, where's this guy's CT scan? So it took a year to do this work and get this picture. So not so easy. Here's the normal lung, and you can see the beautiful alveolar structure, and it really is amazing, the pictures that we've been able to obtain with LA's expert guidance. There's the normal lung here where we nebulized the mouse and then waited 5 days. You can see a lobar pneumonia. Sometimes it's in both lungs, sometimes just in one. Very typical of COVID-19 pneumonia. And if you look at the hemodynamics in these animals, well, the O2 sat in the normal mouse is 95% with a normal 20/60 millimeter of mercury RV pressure and normal histology. And this is a tunnel assay where apoptotic cells show up as green and there's no green in the nucleus. Whereas, and then when we go to hypoxia, the pressure rises slightly, which is hypoxic pulmonary vasoconstriction from 20 to 29. But look at the MHP1 mouse. The O2 sat is 58%, just like our patient. There is no HPV, the PA pressure is around 20. When we give this enhancer of HPV called BK8644, which is the calcium channel activator, we get lots of HPV, so we can restore HPV. And interestingly, the O2 sat appears to rise to 84%. Meanwhile, the infected lung has lots of infiltrates and lots of these tunnel positive. These are airway epithelial cells undergoing apoptosis. So exactly as the theory would have predicted. Induction of apoptosis, suppression of HPV. And I'll just briefly say, I won't go through this paper because I'm over time, but this is a paper from a group that I don't know, but they found what we predicted in humans. So they did hemodynamics of patients with COVID-19 with pneumonia, and they compared them to people that were dyspneic, and they found there was no HPV in these patients. And the only thing I'll just show you briefly is the QSQT in their paper is the measurement of shunting. So there's a lot more shunt in the COVID patients reflecting impaired HPV than in the control patients. And there's very modest elevation of pulmonary artery pressure at 27 millimeters of mercury, but there's no elevation of pulmonary vascular resistance in these patients. So there's no HPV despite their pneumonia. So the summary of my talk is that yes, the virus comes in and replicates, but those same proteins are going and attacking the mitochondria, and they're causing two things: opening the mitochondrial transition pore, causing apoptosis through AIF, and inhibiting complex 1 and impairing HPV. And between the airway epithelial cell damage and the impairment of HPV, you get this situation here, where you've got apoptosis of the airway epithelial cells, and you've got nothing blocking the blood coming into that abnormal lobe, and so you get a lot of fluid flooding. So in conclusion, while vaccination will likely end the current pandemic, this is the third viral pandemic in 2 decades, so we should be prepared for the fourth. MHV1 and SARS-CoV-2 induce apoptosis in airway epithelial cells and impair HPV and pulmonary artery smooth muscle cells. The mitochondria are viral targets and may contribute to the severity of the disease by the mechanisms I've mentioned. Mitochondria may be new therapeutic targets, and improved fundamental knowledge and novel therapeutics are needed to deal with the cardiopulmonary complications of coronavirus.